Hey, this is Joel Duff. Welcome back to my channel, and I got another episode of the Mailbox. This is where I respond to questions that come into my email through Facebook, through my YouTube channel, on chats, on my blog. And today I respond to a question from a friend of mine. How can we know when we look at a strip of DNA, A, T, C's, and G's, which bases are mutations and which ones are the original condition or the ancestral state? Or put another way, or put the way that this person asked me, are a natural feature, or an original feature of the DNA. My answer is really going to be kind of a lesson about mutations and how we infer what are mutations and when mutations have happened to the DNA. So with that, let's get started. Okay, I think I'm more than ready for this because earlier today, I recorded 45 minutes. Actually, I didn't record 45 minutes answering this question because I thought I was recording it, only to discover I hadn't hit the record button. There's nothing more disappointing than going on and on and on and actually thinking, hey, there's some good stuff in here, and then realizing you didn't record it. All right, so second attempt. I have a question that I haven't been able to get an answer to, either from creationists or evolutionists. Is there any way we can tell if something is a mutation or a natural feature? Isn't this just chemicals, CGs, A's, and T's, all right, the nucleotides of DNA? Aren't these just chemicals that are lined up? Is there an objective way to tell if the DNA was mutated in the past? So it's really a question of like, how do we tell what a mutation is, or how can we look at DNA and determine which sites have been mutated and which ones haven't? Notice something in the question, all right? How can we tell if something is a mutation or a natural feature? We'll get to that sort of aspect of this, a natural feature. I'm going to call that the original state or the ancestral state of a DNA sequence. But we'll need to discuss even that. What does it mean, the ancestral state? Right. The way I'm going to approach this is I'm going to talk about a variety of different aspects of mutations. And I'm going to start at, and I'm going to look at different levels of organization. All right. We're going to look at mutations in a single DNA strand. We're going to look at mutations in single-celled organisms and organelles. We'll look at mutations in multicellular organisms. How can we identify mutations in a multicellular system? How can we identify mutations in populations? Then mutations in species, right? See where we're going with this. A little bit about how mutations might add function to organisms. And then we'll even look at mutations in species groups, right? And all along the way, I'm trying to show what are ways that scientists are able to identify mutations that have occurred throughout generations of DNA molecules, whether they be in individual cells, in multicellular organisms, in populations, or in species. We'll begin with maybe the simplest case, because really this question, let's go back to the question real quick. The question is there any way to tell if something is a mutation or a natural feature? Think about this. If it's just A, T, A, C, G, G, T, T, and I just showed you a strip of sequence, and I asked you, which are the original sequences and which are mutations? I mean, stripped of any context, just having ATCs lined up and shown to you, what could you say about that, right? They're all just chemicals, as the questioner points out, right? They're just chemicals. Cytosines, guanines, adenines, and thymines. They don't look different from each other. If I change a guanine into a cytosine, how do I know that it was a cytosine before and now it's a guanine? Because guanines look like all other guanines. There's not like a little marker on them that says like, hey, I once was a cytosine, but now I'm a guanine. Or is there? That's the first thing we're gonna address. All right, so let's keep that in mind. How do we know when we see a mutation, right? What kind of evidence can we bring to bear to make a decision about what bases are mutations and which ones aren't? And a little foreshadowing here. We're going to have some reference points. Relative to what? Relative to the cell, relative to the population, relative to past generations versus present generations, we're going to have some comparative data in order to make this assessment. What bases are mutations and which ones are the original sequence or the original kind of sequence that was there. All right, so back to my outline, but let's go to item number 1. Mutations in a single strand. So this is the easiest way for me to illustrate that we can know, I think very definitively, we can know where some mutations happen because we can see those mutations. And I'll even say that the cell can see mutations. It knows which base is mutated and which one isn't. All right, now what do I mean by that? Well, I mean, right below me here, you have a piece of DNA that's replicating, right? All right, you have a double-stranded DNA. It's being pulled apart, right, into the 2 strands. And then each strand is being observed by DNA polymerase, the enzyme, the machine that copies DNA. And it's going and it's looking for the complementary bases, right? You got an A and it says like, look, we got to slot in a T here. And then we have another A, that's going to be a T. And we have a C, then that's a G, and so forth, right? And that's how it's constructing the other strand. And so if you pull these 2 strands apart, and then you remake each strand using each of the original strands as your reference point, what should you end up with? At the end, you should end up with 2 identical strands of DNA with the same codes. You can take one of those strands, right, put it in a new cell, take the other strand, maybe keep it in the original cell, and now you have 2 copies, 2 cells with 2 identical genomes. But of course, we know this isn't really what happens in nature, right? That's the ideal state. Ideally, that's what would happen. But this is chemistry. Chemistry is not perfect. It makes mistakes, right? Enzymes make mistakes. You got a whole pool of nucleotides that's ready to be used by DNA polymerase. DNA polymerase is zipping down the strand and it's just like plugging stuff in, like, right, A, T, C, G, T, G. And once in a while, of course, it's going to pull out the wrong base pair. It's going to slot it into the right spot. And so you're going to have an A should be paired with a T, but instead it pairs it with a C. Now, clearly that C is a mutation, right? It's not the original base pair that should have been there. Like on the other strand, there was an A and there was a T on the other strand. But this one, when it got copied, it made an A and it made a C. Okay, so now we know that that's where mutations happen, at least point mutations. There's a lot of different, maybe I should back up just real quick. There's a lot of different types of mutations. All mutations are some kind of change in the DNA. That could be individual base pairs that are changing, single nucleotide changes. That could be adding a base pair where it wasn't before. That could be subtracting a base pair, right? Losing a piece of DNA, so that's a deletion. That could be an inversion. That's turning around a section or a segment of DNA, so it's now in the opposite direction than it was before. All those would change the code such that a past generation had this code and a future generation, or descendants of that piece of DNA, now are different than it was before, and we refer to any of those differences as mutations. But let's just concentrate on the single nucleotide changes and how would we identify where single nucleotide changes have occurred in the genome, right? Ultimately, we're going to build up to, I have a genome that I've looked at. How do I know which bases are mutations in that genome? Right now, we're just working with a single strand of DNA in a single cell. How does the cell know, right? Because what the great thing about biology is, great thing about organisms is, they will make mistakes as they copy their DNA. The DNA polymerase will make errors, makes a couple errors every 1,000 base pairs that it throws together. That's a pretty high error rate. In order to reduce the overall error rate of the cell's mechanisms, right, for DNA replication, we have backup processes, we have repair mechanisms, we have enzymes that can scroll down, run down our newly created DNA, observe where mutations have occurred, take out the bad base. So in my example here, we had an A paired with a C, and the C was the wrong base pair. It'll identify that cytosine, excise it, remove it from the double helix, replace it with a T, and you're restored back to your original DNA code. And you should be asking yourself, well, how do they know? How does the cell know? How does the cell know that after it has a double helix, that one strand is the original strand and the other strand is the newly formed strand? Because it has to determine that in order to know which side to remove. If it sees an A and a C, it knows that that's a mismatch. And it's got to fix the mismatch. Well, it could take the C and replace the other side with a G. But no, it usually takes the C and replaces it with a A. Or, I'm sorry, a T. All right, so now you have an AT pair. So it goes back to its original state. How does it know that? Different organisms have different ways of doing this, but the simplest way is to think of it as a form of chemical modification that's being done to the backbone of the DNA. So you can add things to the backbone of the DNA chemically. So you can think of an easiest example of a chemical modification is methylene tags, right? Methylation. And so your original DNA strands, right, have methylation marks or some other chemical marks. There's different marks in some other different types groups of organisms. So just think of it as a chemically altered DNA strand. But when you construct a brand new DNA strand, it has yet to be modified. So there's no chemical tags on that. So the protein structure, the machine that's going to then scan the DNA code, your newly formed code, can look and find mismatches and then be able to see that one is your original strand and one is your brand new strand. And so of course it wants to fix the new strand, not the original strand. And that's your reference point. So this is my simplest example and most clear, most direct example of how you can look at like how a cell actually looks at its DNA and goes like, that is a mutation. Right? Even though it's an A, a C, a G, a T, it's just chemicals. And those chemicals all, there's nothing special about them. They don't look any different than any other ones, but it's their position that's wrong, right? Their position is like they're mismatched. And they can figure out what the mismatch is, like which side is the correct side, which side is the wrong side, and fix the wrong side. And this is awesome because it allows you to go through and fix a whole bunch of the errors you've made in your original DNA polymerase. And it lowers your total error rate so that you will pass fewer mutations next generation. Now, it's not perfect, right? Because you know that, for human beings, we generally pass down around 100 mutations to our offspring. Well, like 50 from me, 50 from my wife. So like I'm making like 50 mutations on average in each one of my sperm that I produce. But that's out of billions of base pairs, right? So that's an extraordinarily low mutation rate overall in terms of DNA replication. So same thing happens in bacteria, right? When bacteria copy themselves, they do have some error control mechanisms. They know which is the new strand, which is the old strand. This can be true they have slightly different mechanisms in different groups, but cells, when they're in the process of replicating and soon after replicating, they have ways of identifying what are the bad bases. And we can see that through experimentation and have actually shown that this happens. All right, so that's like right down at the nitty-gritty level of like, here's the DNA replicating, making mutations, and fixing those mutations. Right, what about mutations in single-celled organisms and organelles? How might we identify that a mutation has happened? Which bases are mutations and which bases are the original sequence, right? The original bases. We have to have a discussion about this original base thing because that's where we're going to get into the creationism sort of evolution thing in terms of different perspectives on that. But for now, let's just work on, I'll try to show you how we infer what mutations are, which bases are mutations. So my simple example here would be, let's think about mitochondrial DNA. All right, so that's an organelle, and that organelle has a genome. The mitochondria has a genome of 16,000 base pairs, a little over 16,000 base pairs in human beings and most mammals around that size. So you've got this circle, 16,000 base pairs, and if I look at my mitochondrial genome and I were to sequence the whole thing, and then I were to look at my sisters, and I were to see that they have like one base pair different than I am. They both have an A at position number 99, and I have a T at position 99. So there's a difference between us. But the question then becomes is, which one's a mutation? Right? Now, why must one of them be a mutation? Because we all got our mitochondrial genome from our mother, because it only passes down from one generation to another through the female line. And so my mom and their mom gave her mitochondrial genome to all three of us. And it should be the same mitochondrial. She has one mitochondrial genome with one sequence. I'm going to disregard the fact for now that she has lots of mitochondria and lots of cells in her body, and there's going to be a few mutations in those. That's the next example for populations, okay? For now, in the sex cells, it's very likely there's any variation there. Probably all sex cells probably have that same, females are very conservative and very careful and have high degree of fidelity, and their error correction stuff is really good in the lines for sex cells. And so my mom likely is giving the same copy, all right, to the kids, except for one of them had a mutation, right? She has one copy she was given. She's probably giving the same copy, except for one of those eggs, there's a mistake made when the genome was copied. And so one of us got that. Well, in the case, maybe I got it because I have the T. And if I just had my sister's A's and my T, but I didn't know anything about my mom's genome, all I would know is probably one of us has a mutation, but I wouldn't know which. In other words, you look at the C, you look at the T, and you'd be like, those are C's, those are T's. There's nothing like I can look at chemically. There's nothing I can do that has a little flag on it in the DNA that says I'm a mutation, right? I can't tell. The only way I can tell is by referencing other genomes and collecting other data. And the data I would need in this case is I would go and sequence my mom's mitochondrial DNA. And if she has an A at that particular position number 99, and then my sisters have an A and I have a T, then my confidence would begin to grow greatly that I have a mutation. I'm the mutant. Okay, I've got a T at that position. And that's not consistent with heredity, with my parent, right? So there was a mutation along the line that got to me. And then if I looked at her mom, right, my grandmother, and I saw that my grandmother also had an A, right, and her grandmother had an A as well, I would see that in the family, an A is passed down at position number 99 throughout the generations, and even to my sisters, but I got a T, right? I think it's pretty reasonable inference at that point that that is a mutation, right? That is a new nucleotide at that particular position, right, within our family. And then I could keep backing up, right? I could look at lots of other relatives, which then would help me to see way back farther in my lineage. So if I looked at a bunch of second cousins and third cousins, and I saw that they all have A's too at position 99, and my mitochondria was pretty conservative, so that might not be too surprising, actually. Now, I go all the way out to second, third cousins, and they all have A's, right? The reason they all have A's is because all of their parents gave them A's, all their mothers and their mothers' mothers and their mothers' mothers. We might be able to trace ourselves back to like great-great-great-great-great-great-great-grandmother who had an A at that particular position, and all the descendants have an A, and eventually one of us doesn't have an A. Right? The simplest explanation, the most parsimonious explanation, using Occam's razor, right? We would conclude that I have the mutant, that my T is a mutation. All right. So even though I didn't actually see the T happen, right? I don't have an eyewitness record of the actual event, seeing that DNA replicating, and then I saw the wrong base pair inserted, and the DNA replicate, and the DNA error correcting mechanisms didn't catch it, and it got passed through. Once you have an error and it copies itself again, then the error basically gets fixed because our bodies can't tell that those are mutations at that point. I probably should have said that earlier, but that's the thing about mutations is at first they make kinks in the DNA, right? An A and a C doesn't quite really line up well. The bonds don't really work that well. So you create a an abnormality in the double helix. This is one of the ways you recognize is that your body can recognize there's a problem here, right? Let's look at this site that has a kink in your DNA and figure out what's the wrong base pair and then remove it and put the right one in. But once your DNA goes through another cell division and divides again, well, guess what? You're going to pull those two strands apart, and then you're probably going to copy, you had an A and a C paired. Now you're going to have an A and a T, and you're going to have a C and a G. And the C and the G are going to be totally wrong. That has nothing to do with the original generation. Now, if you go back 2 generations, it was an A and a T, but now you have a C and a G in one of the cells. The other cell, the A pairs with the T, and you actually go back to the original state. So one cell gets the original, is sort of back to normal. And the other cell that gets the other strand of DNA gets a completely wrong base pair. And now we'd say that mutation is fixed. Because once it's C and G, yeah, the cell has no way of looking at that and going like I can tell that's a mutation. It doesn't know at that point. It doesn't matter if one side is methylly marked or something like that, you know that's the original strand, because the other strand has the complement to it that looks totally normal. And so no error correcting mechanism is going to be able to go in and say, like, we need to take both the C and G out and replace them with an A and T because that is the original state, right? The natural condition of this particular piece of DNA. Okay. All right. So back to my single-celled. So organelles, that's an easy example because, like I was saying, in my family, extended family and extended family, if everybody has A's and I have a T, then obviously I have an error. Then we could go back and we look at hundreds of thousands of genomes, of mitochondrial genomes, and we find out like maybe everybody in the human race has an A at position 99. And I'm the only one that has a T. It's pretty apparent that I have a mutation, all right? Now, it could be that 40% of the population has an A, all right? And 60% of the population has a T. Or let's say it has a G, right? Well, that means that somewhere either the A or the G is a mutation, right? And then my T is yet another further mutation on the A, right? The A might be the original state, and then I turned it into a T. Maybe somewhere else farther back, like someone's great-great-great-great-great-great-grandmother, where we're related to, they had 2 children, and she gave an A to one of them, and she gave a G to the other one. And that G then got propagated out into millions of people, and the A got propagated out into millions of people, and eventually I had a T, right? So the more data I have, the more confident I can be about the origin of different mutations. So now let's take this a little further. The mitochondrial DNA, if we can all trace our history back to that mitochondrial Eve, right? But in evolutionary biology and for creationists, there's still a mitochondrial Eve. There is a person in history, right? There is a person in history, in human history somewhere, that had a mitochondrial genome, all right? And so it had one sequence. And at position 99, they could only have one particular base at that particular position, an A, in which case they were an A and a T, right? Paired. But we're just referring to one strand. They had an A. And then their offspring represent everybody that's alive today. Right? All the other lineages go extinct at some point, and so we're all related to one individual. And in a young Earth creationist model, and in, well, not even young Earth creationist model, but even other Christian models of history where you have a sole female origin, all right? Eve, the literal Eve, right? She has one mitochondrial genome. And so she's passing that down to multiple offspring. And she might make a mistake even in that first generation. Maybe not. Maybe all of them got a faithful A at position 99. But then at some point, some generation, some offspring is going to get a different base pair. And so you're going to end up with multiple different alleles, variants in the human population. It can be a little difficult to know what the original base pair was. I mean, if you had 50-50, like 50 A's, 50% were T's, you'd be like, I don't know. I can't. That's where you'd say, I don't really know what the original state was. I don't really know what the original base pair was at position 99. It could have been an A or it could have been a G. Could have been an A, it could have been a T. Unless you have another reference point, right? Something else to compare outside of that group. So you can collect a larger data set and then put all of the current data into that context. What would I mean by that? Well, that would be if you thought that human beings were a population that was related to something else, like chimpanzees or something like that. You could start looking at all the primates, and you could look at their mitochondrial genome, because they have mitochondrial genomes about the same size, and it has a base pair number 99. And you could line up the sequence of humans and all other primates, and you could look at position number 99 as a homologous site, the same exact location. It's at the same position in the same gene, and it's base pair number 23 in the first gene, right? And you could look at that position and ask, like, what do all the other primates have? Especially, say, chimpanzees and great apes, or gorillas. And if you saw that they all had A's rather than T's, and then you look at the human population and like half of them have A's and half have T's, that would be, you could infer, if your model was that those organisms are related by common ancestry, you would infer that since they represent the broader group of apes and they all have an A and they share an ancestor, they're all sharing the A's. And then there was one group, one population that had A's, and eventually there was a mutation creating T's, so that that particular population, the population of human beings, has A's and T's. In which case you then can look at the T and you can say, like, that's the mutation. Right? And then if you see a subset, a small subset of humans, like my family, and instead of having a T, they have a C. Then you say, well, that's a T that mutated in their they came from T's, which was a mutation, but then their T changed to a C, so that's a further mutation at that particular location. All right, my other example, I'm talking too long about this. I'm already, as I look at my clock, I'm 30 minutes in already. And we're only on item number 2. But single-celled organisms, the other example that is clear, a very clear example of where we know where mutations are, because we've done the experiment, would be like Richard Lenski's work in with bacteria, right? E. coli, right? And he took a single bacteria, all right, selected a single bacteria, grew that into a colony, separated that into separate individual starting points, and then began to grow those colonies for, now 80,000 generations. So you started out with a single bacterial genome that had a single sequence, right? Every single position, there could only be one base there. At base pair position number 5,000, there could only be an A, a C, or a G, or a T in the original state. We'll call that the natural condition, right? The original state. And over time, what happened, of course, is each of those different colonies in separate isolated groups that are being replicated, they each had mutations, right? And they accumulated mutations over time. And so some of those base pairs, base pair number 5,000, in one of those lineages might have mutated. And so if you sequence the original lineage, which they've done because they have the original lineage, and then you sequence 5,000 generations later, 10,000 generations later, 50,000 generations later, 75,000 generations later, and you look at the genome, you can see all the locations that are different than the previous time you sequenced the genome. And since you know they are in a linear set of generations, you can know for certain, right, at least a high degree of probability. Right? I mean, I suppose there could be contaminants and if some of the things, but you can be pretty sure that if there's an A there now where it used to be a T thousands of generations earlier, that a mutation happens somewhere between that 500 generation and 50,000th generation, right? You know that mutation has occurred because you know the original sequence and it didn't exist then. So in that case, you can look at a genome, the genome at the 75,000th generation, and you can just go across the entire genome and you can go like, mutation, 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 point mutation, deletion, insertion, inversion, duplication. This whole segment has been duplicated, so now there's 2 copies of it. That's what happened with the CGAT gene. That's a duplication of a segment of the chromosome. which then allows the one lineage of those E. coli to behave differently than the other lineages. All right. So they added a new characteristic to their population, to their and changed their survivorship, right? Changed their fitness. And we know that that is a duplication because we have the original sequence and it didn't have that duplication. So we don't have to have actually seen the duplication happen to be quite certain that it did happen in that case and identify it. All right, let's move on because we got to get to, I think, what is more interesting territory. But we have to build, I'm just building the case that there are times where we are very certain about that's a mutation, that isn't a mutated spot. And now I'm going to get to times where we're not, it's not as obvious, but we still have ways of inferring mutations. So what about mutations in multicellular organisms, right? You and I, we started as single cells. We have 2 copies of our genome, but let's just focus on 1 copy, like chromosome number 1, copy number 1 from mom. Right? As we create a trillion cells, we are going to make mistakes every single time we copy chromosome number 1. So in my body, in this finger compared to this finger, right? If I looked at the entire 100 million base pairs of chromosome number 1, I expect there to be thousands of differences between one cell in this finger and a cell in this finger because those cells are the result of thousands of generations of replication in order to produce those cells, especially if they're in my epidermal tissue, because those are constantly replicating in order to create more skin. All right, so they've gone through a lot of replications, each replication is producing potentially hundreds of mutations. They're not as faithful. We're not as faithful with our DNA replication as we are with our sex cells. And we spend more time protecting them than we do our basic cells because they're expendable, right? I'm going to die. And it's really only the sex cells that you want to preserve and keep the genome better for future generations. All right, so the point is, I could look at my original genome. And the way I can't go back to my first cell and sequence it, but what I can do is I can sequence my parents. Right? I can sequence my parents, and I can see for every single position on my chromosomes, what is the probable sequence they gave me, right? They made a couple mistakes when they made me because we all get 50 to 100 new mutations, when we're born. So there's going to be some mutations that occur, but it's going to be a very, very small number compared to the billion base pairs we have. And so I could probably create a fairly faithful representation of like what my original genome looked like. And then if I sequenced my finger and then I compared the two, well, it's the same thing as the mitochondrial DNA or the examples I just gave before, right? I could see like all the positions that have changed since then. And I could identify all those as mutations, right? But only because I have a reference point. I have something to compare it to, a before and after shot. And that's the theme here. You have to have the before and the after, and you have to have maybe multiple data points in order for you to create a pattern that allows you then to identify the most consistent explanation for the pattern you see is this was a mutation. All right, so multicellular organisms have an entire lineage of mutations in their own body, and there are some really interesting papers out there, like actually tracing the history of a cancer, showing early stages versus later stages, and you can actually see the mutations increasing and changing and morphing the cells, so you know all the mutations, and we're trying to figure out what all those mutations do. And so we can identify like all those mutations that way. But let's get on to populations. All right, so I can't remember where I put my notes. So I'm flying blind right now because I had written down some examples earlier today during my first attempt to record this. Okay, so now we come to mutations within a population. And so now we have a whole bunch of individuals. And we have a bunch of variation in that population. And we look at that variation, again, we're just going to work with individual mutations, like individual sites. A's, T, C's, and G's are changing to different A's, T, C's, and G's. And we look across that population, we'll see that some individuals have an A, some individuals have a C, maybe some individuals have a T, some individuals even might have a G at any one particular location in the genome. And so the question would come up then, which is the original base pair? Right? Which is the A, the C, the G, or the T the original condition, the original state. Now, I guess maybe I should take a small tangent here and say that going back to the question says, it references asking creationists and evolutionists about how we'd identify mutations. And so I want to address that. We're going to get more toward that in a few minutes. So if you come from a creationist perspective, and let's say a young earth creationist perspective, where God is, in their model, God is making a bunch of different separate kinds. And so there is at some point the instantaneous creation of the entire genome of, say, human beings. So there isn't anything before that point. In other words, there's no reference point. There's nothing to compare a human to in order to tell what's the original state. Because that's what I'm kind of emphasizing. If you have an A, a C, a G, or a T, and some individuals have each one of those, and you ask, what was the original state? It can be a little hard to tell. In fact, it might be impossible to tell, right? In the situation where there was no other ancestor to go back to. There's no history before that. And so let's play out a couple scenarios so we can see how this would work. Let's take lactose tolerance. So I want to give you an example of where you can, even in the human population, come to a pretty reasonable conclusion about lactose tolerance mutation. So lactose tolerance is in most cases, is a single mutation, single base pair change that changes a basically developmental switch. So turn something off at a different time. Different timing. So allowing for you to continue to be tolerant of lactose later in your life, because really everyone's tolerant when they're first born, and then they lose tolerance. So there has to be a genetic change in order for you to create the signal to say, like, you know what? Don't lose don't lose that tolerance. Just keep on tolerating milk for your whole life. Now, let's just say that lactose tolerance is, so it's a change in the genome, right? It seems to be a mutation. Now, you might argue, maybe that was in the original condition, like lactose tolerance. Maybe Adam was lactose tolerant and Eve was lactose intolerant. One of them had one variation, the other one had a different variation of this gene. Right? And then they had offspring and they've shared it with them. And those offspring then had offspring and so forth. And some offspring ended up with 2 copies of the lactose tolerance and other ones ended up with 2 copies of lactose intolerance and so forth. And you ended up with individual subpopulations that were less tolerant and other ones that were tolerant of lactose. Right? I guess that would be possible, but that doesn't actually fit the observation. The observation is that there are large swaths of the world that are intolerant. There are large populations that are generally tolerant. And when we trace back the history of human populations, it seems like lactose tolerance kind of quickly funnels down to a couple different individual locations in more relatively recent history. Right? So human beings seem to be much older if you look at the wide swath of all human history captured by all people alive today. And I guess you could say even archaic, Neanderthal sequences that we have. Lactose tolerance looks like a subset of all those. But here's the other thing about lactose tolerance. It's not all caused by the same mutation. There is several origins of lactose tolerance, and there's different mutations that cause lactose tolerance. So if you're lactose tolerant, you can trace yourself back to a particular mutation, but somebody else might have a different mutation and they can trace all their relatives back to another location, both locations, I believe, in Africa, in which it appears that there was an individual in those 2 locations at different times, not exactly the same time, in which there's an individual that had a mutation, like probably in the sperm or the egg, there was a mutation in that particular spot in the genome. That individual was tolerant to lactose. And that might not have made any difference to them because it's not like, oh, they knew they were tolerant and they started drinking milk and it was some huge advantage. It could just be that it was neutral. And they had offspring and they passed that on. So now there's like several members of that individual little tribe that are tolerant. And then later on, I'm going too far into the story, but later on, as more of them are tolerant, but maybe not really using the gene, so it's not being selected for. You know, they've got goats and, you know, they're drinking milk, giving it to their young, and then they're like, well, yeah, but our kids are still like able to drink it, so we just keep drinking it, and then others start drinking it. Then it becomes an advantage, right? And there's enough individuals that have the allele that enough of them have that advantage, becomes a thing, right? This seems to have happened at least twice in history. Now, if it happened twice or even 3 times, it gets harder to explain all of those things as being just original variants in, say, Adam and Eve, right? It's easier, it's more parsimonious to say that the original condition of human beings is lactose intolerance, right? That seems to be, if you go back in time, there seems to be a time in which everybody was lactose intolerant. And then there was little pockets, little places in time where lactose tolerance seems to have suddenly sprung up. And then it spreads from there and gets passed on to a whole lot of people, right? And it's, I guess you could say it's gradually spreading further, because being able to drink milk has been a pretty good advantage for most people. Right, so that's where I'd say that's where you're inferring. You're saying, I didn't actually see the mutation, but this appears to be a subset of an original condition. So this seems to be the mutation, right? Blue eyes is the same way, right? Blue eyes is actually created multiple times as well, or formed multiple times as well in different ways. And so you could look at a whole bunch of things, and we could look at even more recent mutations in the human condition that you wouldn't trace all the way back to, say, a common ancestor. So there are mutations of which, like, only people in some small community in the Swiss Alps or something like that, in a village, like half the people in that particular village have a particular allele, variant of a gene, and nobody else on the entire face of the earth has it. The most likely reason for that is because they experienced a mutation. Some ancestor experienced it and have passed it just to individuals within that particular population. So that's the same thing as going back to single-celled organisms or a multicellular situation where like my whole arm, right, has a unique mutation in all the cells in that one arm compared to the rest of my body. How would I explain that? Well, probably in early in development, I had a cell that was really responsible for dividing and becoming the majority of that particular arm. And it had a mutation and then passed it to all of its descendants. And those descendants made my arm. So therefore, this arm has a unique mutation compared to the rest of my body. And so again, it's comparing. I couldn't tell if I just took that one cell out and I had nothing to compare it to. I would never know that that particular spot was a mutation. It just would blend in with all the rest of the sequence, and I really wouldn't have any way of being able to determine, like, oh, that thing right there is not normal, right? And so, but by comparing it to the rest of my cells, and especially if I went and compared it to my parents, I would realize that's a mutation. So I think that gives a couple examples of how we would identify individual variants in a population as to their origin. Is their origin as a mutation, or is their origin as the original state for that particular population? Again, everything is a reference point to something. If I reference the population could be the whole species, right? The whole species has a particular original base pair at a particular position. And then within the population, the variations are the mutations, right? But how would I know what the original state is? I probably have to compare it to other populations in order to know, like, oh, all these other populations have an A. That's probably the starting condition of this population because it branched off from those populations. And then all the other variants inside that are different than A are actually mutations. All right, what about mutations in species? So we're getting really close to that before, where I said, like, a population might represent the whole species. Well, now let's think about coyotes and dogs, right? Or coyotes and wolves. Coyotes and wolves, if I compare their genomes, there are clearly differences, even if I compare lots of coyotes and lots of wolves, right? I have many sequences from many wolves and many coyotes, so I kind of have a snapshot of their overall genetic variation. I would probably be able to pick out hundreds and hundreds and hundreds of locations where every single wolf was different than the coyotes, right? The coyotes were all the same. Base pair number 99 was a T in every single coyote I look at. And then I look at the wolves and I see like base pair number 99 on chromosome number 1 is an A for every single wolf. Right? So I'd say we call those are fixed variants between different species. Some variants are going to be shared between different species. Like most coyotes have A's, most wolves have T's at some position, but some coyotes have A's and some wolves have T's at that particular position. But there's an overall frequency difference between the two, which can add up to actual differences between averages between species. But let's just focus on the fixed differences. Like this is always different in this species versus this one. There's going to be thousands of sites like that between a lot of species. You would then ask, which is the mutation? Is it the A in a coyote or is it the T in a wolf? I don't remember which one I used A and T for, but now they're A's in coyotes and T's in wolves. Which is the original state for that particular location in the genome? How would you tell? Well, probably going to need some other further reference point, right? So if they're all A's, all T's, and we think that coyotes and wolves have a common ancestor, and evolutionists and young earth creationists both believe that, right? Ken Ham believes that coyotes and wolves are the same kind of organism. They share a common ancestor in the past. They were not separately created genomes for a wolf and a coyote. So therefore, God didn't just like make an A at that position and put it in all the coyotes and make a T and put it into the genome of all the wolves. No. In the young earth creationist model, God made some common ancestor to coyotes and wolves. In fact, he made a common ancestor to all canines, according to most young earth creationists. So lots of different species. So we can determine whether A or T is the original state, all right, the original condition. Even if original condition, what we mean is what did God make in the original genome? And here's how you do it. The same way we did it for everything above, right? That's if we're going through the same pattern. I would sequence African wild dogs and other related species, other things we know are similar to coyotes and wolves. In other words, the outgroups, right? That's what we call it in phylogenetic terminology, parlance, right? We would look at the other kinds of canines. So now let's say if I looked at a bunch of other wolf-like species, let's forget the foxes for now. We look at the other wolf-like species that are most similar to coyotes and wolves. And I look at position number 99 on chromosome number 1, and I find out that all the other wolves have an A, right? And let's say that wolves, all right, the actual wolves, Canis lupus, has an A there, and they all have A's. But Canis latrans, the coyote, has a T. Now the T is an outlier, right? It looks like a subset in a much larger analysis in which all these other species all have A's. And then if I want to go look at foxes and they also have an A at position 99, then we're saying like all canines have A's except for this little coyote over here population. That's kind of like saying I have a T in my mitochondrial genome, or just my family has T's. My mom and my two sisters and I, we all have a T at position 99 or mitochondrial genome. But if I go back in time and I look at everyone else that's related to me, they all have A's. Well, then we're the mutants, right? That's the easiest explanation. I mean, it's really hard to turn that around and go the other direction. It would be possible, but it's certainly not the most simple explanation. It's going to require a lot of ad hoc throw-ins to try to explain it in any other way. So I hope that's obvious. Like, right? The T then sure looks like it's a mutant, right? Sometime when the population of coyotes and wolves, there was an individual that had a T, right? Compared to the A that all of the canines have ever had before. And that T they then passed it on to a few offspring. And maybe a couple of those offspring Like they travel, as coyotes, as wolves are known to do, they travel some distance. They get separated from the rest of the population. They reproduce with each other, which increases the chance they're going to keep sharing that T, right? And eventually it fixes in the population, right? Fixes means like every individual, all their 99th positions of the chromosome number 1 in all individuals all have a T there. So now it is truly different than the wolves. And then that isolated population will undergo a lot of other changes and eventually become a new species, right? And so now the species have a fixed difference between the two. And we can be pretty sure that the T is a mutation and the A is the original condition. And if you want to go back to Noah's Ark and say like there was 2 canines on the Ark, you could be fairly certain in your prediction that those 2 canines probably had an A at that position. It's extremely unlikely they were hybrids and they had an A and a T. Because then you'd have to argue that when they got off the Ark and they spread A's and T's, because they'll have offspring and they'll have a mixture of A's and T's, and almost like at least 25% of the population would be A's, would have A's. Right. And as they separate into many, many different species, they're going to carry the A's and the T's into all those species. And that's going to be variation within all those species. But if you already tested all those different dogs and they all have an A, well, that's a highly unlikely scenario that you would have lost the T's in every single species. That's hard to do, especially when you get populations to relatively high levels and there's no selection. It's just genetic drift. I mean, that's virtually impossible to get rid of the T's by accident. And somehow only coyotes got T's? That wouldn't really make a whole lot of sense. It's so much simpler to say that there was originally A's and then there was a mutation and now you have a T. All right, so this whole idea of created heterozygosity, it's problematic for a lot of different ways, but you might be tempted to say, like, I'm going to take all the variations I see among all the different kinds of, all the different species of a kind of organism, like canines. I'm going to take all the variations they have, and I'm just going to squish them into 2 individuals. If you play that out in terms of how those alleles would spread and propagate, you wouldn't get the patterns that we observe today, right? That's not the predicted pattern you would get. It's much simpler, and actually it fits the data much better. It fits our actual observations much better. That what you had new mutations. Lots and lots of the variation in species today are new mutations that occurred within those populations, right? Rather than being shared by some common ancestor. All right, so I'll go one step further with this. I was just looking at cheetahs because I just talked about cheetahs the other day. I never quite got to the genome portion of talking about cheetahs. And in cheetahs, there's almost no variation, right? They're the weird example where they've been through some kind of bottleneck, kind of like Noah sled, except that wouldn't work in the young Earth creationist model for cheetahs. Cheetahs seem to have been squished down to just a few individuals or a very small population, and therefore they all bred with each other, and so they don't have much variation at all. But what's interesting about cheetahs, and really all the cats, and this is going to be true for dogs too, is when they looked at the entire genome compared to all the other cat species. Cheetahs have I can't remember if it was 59 or 79 different families, or we'll just say genes, 59 genes that no other cats have, right? 59 coding sequences that code for some kind of product that other cats don't have. So where did those come from? Now, the lion also has genes that other cats don't have. And your house cat, or the sand cat, it has genes, dozens of genes that no other cats have. So there's 2 ways to explain that. All right, 2 possible ways to explain this. One is the 2 cats that were on Noah's Ark had incredible amounts of variation. They had huge numbers of genes. And then as they went out and they reproduced and they formed separate populations, they had mutations, right, that knocked out genes in some populations. And in another population, it didn't get knocked out. And so you end up with a cheetah that has 59 genes that didn't get knocked out, but it got knocked out in all the other species, right? All those other species lost the ability to use that particular gene that was in their common ancestor on Noah's Ark, and by inference would also be the common ancestor all the way back to creation. God made the original feline with, all these genes, but now every species that exists today only has a subset of them. Now, it turns out most of the cats have basically the same length of chromosomes. They have the same amount of DNA, similar sized genomes. They have different numbers of chromosomes, but their overall genome's around the same size. So they actually have the sequence where the gene is like, so a cheetah has a gene at a particular location on chromosome number 1. And if you go look at a lion, they have an area in their genome where that gene could exist. Like it's between these two genes, which we both share. And in between, cheetahs have a gene and they actually make a product there. But in the lion, you look at it like, it doesn't really make anything, right? It's just nonsense sequence. And so how did that become nonsense? Because of mutations. A mutation occurred and broke that gene. And then more mutations happen in that particular spot. And they start to accumulate in that particular location. And pretty soon that sequence is completely non-functional and very different than the cheetah sequence. All right, so there's another way you can look at a genome and say to yourself, even from the young Earth creationist perspective, you can look at that genome and say, the functional gene, I'm just speaking about young Earth creationists now. In a young Earth creationist model, every gene was in the original, created state, right? Every gene existed in some original genome of some original feline. And then some of these genes had mutations. And so if you see a broken gene and you go into the gene, you say, I can see where there was a start codon, but there's a broken stop codon, and there's a bunch of other problems. Each one of those each one of those spots is different than the gene in the cheetah is probably a mutation, right? You can infer that those sites are mutations with high confidence for young Earth creationists. Now, for a evolutionary biologist, they could come out that's in two different ways. There's two different possibilities, and it might be hard to tell which is which. One would be that you had an original feline, because young Earth creationists and evolutionary biologists both agree there was an original kind of feline, an original ancestor that then had offspring that resulted in the separation of populations into dozens, actually 100 or more different species that have, wildly different characteristics, but still share that common ancestor. And so from an evolutionary point of view, you could say maybe one of those genes was in the original ancestor. And then as it got passed down, it mutated in some lineage, and in some of the lineage it didn't mutate. And then that lineage then turned into a whole bunch of different species of cats, and they just shared that mutation, and they still had that broken gene, and therefore they all can't use that particular gene. And this other lineage kept that particular gene and was using it because they're in a different environment, maybe it's useful. So selection is actually saying, hey, don't mess with this thing. And they end up using it, but maybe only cheetahs do. Maybe it's the reason why cheetahs can be run so fast. It changes their muscle structure just slightly. And so you'd look at that and you say well, maybe the cheetah is the original version of the gene, right? And all the others are the mutants. There's another possibility though. Since the cheetah is the only one that has 79 different genes, it is possible that those genes didn't exist in the original ancestor. Right? And that at some point during the evolution of that lineage to make cheetahs, right, they've had mutations that, I should bring up the next one, they had mutations that added new function. They had mutations at a location that actually created a protein coding sequence. So they took a sequence that was out in no man's land between two genes, and there was a mutation that created a start codon. And that start codon then made a protein, right? We know this happens, and we've seen this actually happen, and we can show it theoretically happening as well in many different organisms by comparing genomes. That mutations can create start codons, stop codons, and then the cell itself will just start making transcripts and then start reading those codes and making proteins. And 99% of the time, the proteins are nonsense, right? They don't really help the organism, but once in a while they do. And so you have de novo creation of genes. So that's another possibility. You would have, those are the mutants. The mutants are actually things that make things that are like new properties. So it may be tempting to look at it and say, like, that's functional, so therefore it's the original state. But actually functioning might be the mutation. Right? And the way to tell that would be, you would look at all cats, and only one cat species has a functioning version of this particular portion of the genome. And then the evolutionary biologist has another advantage here. They can look at all carnivores. And they could look at the same area of the genome in all carnivores. So like you got gene A, you have gene B, and between gene A and B, one cat species has gene X. All the other cats have a space there between those two genes, but they don't make anything. And then you go look at dogs and hyenas and wolverines and a bunch of other different carnivores, and you look at that same area of the genome, and you're like, you know, that's interesting. All of them have a same gap, but none of them also make anything there. They don't actually use that for anything. All right. They're not transcribing it. They're not translating it. We can actually show that they're not using it in that way. But the cheetah is. That increases the odds that the cheetah, being an oddball, is actually the one that found a use for that particular location. And the way they found a use for it is they had mutations, and some mutation hit the magic bullet in terms of like turning on that particular location and making a product. And natural selection then sculpted that from there on. It really, all it had to have was just like a tiny bit of use, right? And so it wasn't negative, might have had been slightly beneficial. And then if it has new mutations, it will choose the ones that make it even more beneficial. So that gene can become more and more beneficial over time by adding new mutations over time. To the point where today it looks like a significantly different gene that has a really important function. But it didn't necessarily have to start that way. No one's saying that a new gene, de novo gene, is just like all of a sudden, oh, I'm like an integral part of this organism. No, it's usually like I barely do anything and I'm just kind of hanging around, and I get copied into multiple different cheetahs, and then they start making more mistakes. And once in a while I get a few new mistakes in that particular segment that make me even more beneficial. And then I kind of catch on like wildfire over time. It's like, as I go through there. All right, so that's what I mean by mutations adding function. So now we have an interesting interplay here. We've got, now we have the whole creationist idea of genetic entropy that comes into play. So how would you identify mutations in a young Earth creationist model? Well, they say like the original genome had no defects, although what a defect is and what it isn't is really hard to define. I mean, the same question could be asked of them. If I just looked at A, C, G, T, G, G, I just gave you a piece of code and I said, is this a perfect sequence? Is this an original God-created sequence versus another sequence that's A, C, G, T, C, G, C, G, and it has a few differences? And I told you, can you pick out the original version, like the perfect sequence? God's original plan versus these are mutated? Of course you can't, because they're just A, T, C's, and G's. You have no reference point. You'd have to know, oh, you're showing me a sequence from the original organism in its original state, like on the day of creation. Then you might say, well, that was God's original sequence he made, right, for that genome. And then you show me today's descendant and you see the differences. Then you would say, those are mutations. And if you want to invoke something like genetic entropy and add up the total number of mutations, like, oh, here's all these mutations, and and you could ask the question, what are they doing? They're causing all these problems, right? But in order to know that, you'd have to know what the original condition was. This is one of the things with genetic entropy. You're assuming you know what the original state is. And so you can look at an organism's genome today and point to all these different issues and say those are all mutations. I mean, maybe the original state was you're not using this portion of DNA for something, and a later mutation is that you are using that. Maybe that was God's plan. Right? I mean, there are models in young Earth creationism where, like mediated creation, where God knows that in the original genome, you don't need to have a program to make a spine or a thorn or a nauseous chemical to ward off prey, right? And so those don't even need to exist originally. So God's plan could be to know that there's going to be mutations that are going to change some of those sequences into sequences that actually form into different proteins than they originally were. And those different proteins will then cause different phenotypes in that organism later on. So what you think of as the perfect sequence originally, the mutations actually are what making that organism adapted to a new world, right? And so without those adaptations, actually that organism would die and wouldn't survive. Right? So there's all kinds of weird ways to think about mutations in this fashion. So actually, we've already been talking about mutations in species groups, because I've mentioned in order to really identify the original state of a particular base pair in a DNA sequence, you have to compare a lot of different individuals outside of the specific group or the specific mutation you're looking at. You have to look at a lot of individuals to see where the mutations are. You see where they're spread out, how they're spread within a population. And then to know which is the original state for that, you have to look at broad populations outside of it. You know are related to that population, but haven't had direct contact with them for a long time. So in other words, you're going back to a common ancestor, because everything depends on that common ancestor. You're always reading a sequence through the common ancestor, because mutations are all made by an individual somewhere, someplace in time. And so you have to be able to identify, if I go back in time a certain distance, at some point, the mutation should disappear, right? Because it has an origin point. And so the farther back you can infer in time, the greater chance you have of identifying the mutations versus the original DNA condition. Now, from an evolutionary perspective, saying ancestral state is a relative thing. The ancestor of what? The ancestor of carnivores? Right? If I want to know what the ancestral DNA sequence is of all carnivores, then I'm going to sequence all living carnivores. And I'm going to say, look, almost every carnivore has this particular gene at this particular location and has this particular sequence. And here's an outlier. So the ancestral condition almost certainly is the one that is the majority of what all the descendants are. But even then, I might not be certain. So what I could do is then I could compare my carnivore predicted ancestral sequence to other things, other families of mammals that are similar to carnivores. And if I found out, all the other things around it, related to it, all have an A at position 99, and most of the carnivores have an A, then I'm going to conclude that the T is the mutation within the carnivores. Right? So it's all looking at that ancestral. If I want to know what the ancestor is, I typically have to have something outside the ancestor. So I'm looking at a deeper node. And as long as I can look at a deeper node, I can infer underneath for more recent nodes, what are the mutations and what aren't the mutations, just like I can in human beings. All right, for mitochondrial DNA or Y chromosomes or all that, we can infer back, because we're always looking at ancestors, deeper ancestors, and so forth. But if you want to know the original code of the Y chromosome, that's kind of hard to predict from a young Earth creationist perspective, since you don't have anything to compare it to. You can't look at a deeper node. You can't look at like, okay, great apes, here's their average sequence. So therefore, human beings must have started with that particular sort of average sequence, typical sequence. And then any changes within humans are mutations relative to that. All right, what else did I want to say about this? I feel like I had a couple more points to make, but let's go back. But before I leave, let's go back to the question. Let me make sure that I've actually addressed the question, because I have to remember what it was now. I have a question, haven't been able to get an answer from either creationist or evolutionist. Is there any way we can tell if something is a mutation or a natural feature? Now, see, I think the person asking a question here, when they say natural feature, Natural would mean, in my mind, they mean the original feature. What was the, because we ask these questions all the time in young earth creationist and evolutionary conversations, where it's like, what was the original condition of a carnivore? Were they all meat eating? Well, young earth creationists say, well, they couldn't have been meat eating because in their minds, there was no carnivory in the original creation. God made them not carnivores. So then you start asking questions about, well, what about the features that make something a carnivore? Where do they come from? So they were either like not used for carnivory, like sharp teeth, or they came into existence later. And how would they have come into existence later? Maybe through mutations, right? Maybe through genetic modifications somehow, or pre-programmed genes that were turned on later by some other method that we don't know of yet. Right? These are all kind of ways that young earth creationists try to understand it. But what they're asking is, what they're fundamentally trying to do is figure out what was the original created kind, the original created condition for all these different organisms, their phenotypes. But in this question, we're asking, what's the original DNA strand? What's the original code for every different kind of organism on Earth? For an evolutionary biologist, they might be interested in what's the original code in the first cell that was like a living cell, right? What was its original code? And then everything after that is a deviation of that code. Just like everything after the two dogs on the ark, everything that you see in canines today is a deviation on that particular theme of that genome. There are tens of millions of new variations within the carnivore kind that I'm sure didn't exist in the original carnivores that were on Noah's Ark. And therefore, they're all changing those organisms. All right, so I think that's what's meant here by natural feature. And so the idea that somehow we could look at DNA and be able to say, like, base pair number 1, 5, 7, 15, 24, 32, 64, those are all mutations. And all the other bases are exactly the way they were at the very beginning. Like never was a base before that because they don't have an ancestor, right? So what the original organism's DNA is. From an evolutionary perspective, it's hard to think that way because every single base pair in the genome is likely different than it was in an ancestor if you keep going farther back, right? Every single base pair has been modified somewhat. Now, there are some core genes. There are core genes and core sequences that make up products that every single living thing has. Like, there's ribosomal RNA, there are ribosomal proteins, there are certain like core elements of the basic operating system of any living thing. And they have genes for which there are particular sequences in those genes that may have never changed. So in that case, actually, there are scientists, I'm sorry, going on to the tangent, but I think it's relevant. There are scientists who have spent a lot of time looking for those core sequences. And the way they do that, how do they do it? They sequence genomes from, and this was started along by Carl Woese and others. This is how we came up with the original tree of life. They found genes for which every living thing shares, and they found out that every living thing actually shares some sequence in those genes. Now, there is some variation in that gene, and that's how you can actually say, like, plants have a core set of sequence that's identical to mammals, but there are these differences, and that's what makes plants different than animals, and animals are more similar to each other and plants are more similar to each other. And so you can look at, like in bacteria and other things, you can sequence hundreds of thousands of genomes, line them all up, figure out the genes that are shared by every organism, then look at the sequences of those genes, and you can find locations where it's like these particular base pairs have never been different in any organism we've ever looked at, in which case you would infer that those are the original, like they represent some ancestral sequence state. An ancestral sequence state that has never been altered or changed since. Doesn't mean they haven't experienced mutations, but any mutation that happens there, the organism immediately dies and so is removed from the population. So every survivor that has ever had more offspring has never had a mutation at that particular spot. And so there you're really doing some deep inference of finding the natural original state of the sequence of those genes. All right, so is what I've done been objective? Yeah, I think it's objective. Now, objective can mean there are some assumptions, but in many cases, the assumptions of creationists and evolutionists are the same for determining how mutations could be objectively identified. All right, let's quit there. Hey, thanks. And thanks to the person who asked this question. I think it's a really great question. It's a really good thing to think about. I'm sure there was a whole bunch of different things that as soon as I stop this recording, I'm going to think, oh yeah, I should have mentioned these 10 other things. Really cool examples. But I hope that gives you some idea, starting from the simplest DNA strand all the way up to looking at populations and species, how biologists, geneticists can look at sequences and determine what are more recent elements, new versions of sequences versus ancient sequences that haven't changed over long periods of time. All right, let's quit there. Thanks for listening. Subscribe, like, all that good stuff, and we'll talk to you later. Bye-bye.